Okay, thank you. So my name's Dr. Claudine Kraan. I'm from the Diagnosis and Development Group at the Murdoch Children's Research Institute. And thank you for having me today. So today I'm going to talk to you about gait in Prader-Willi syndrome. What is gait analysis? So basically, it's the breakdown of the gait cycle into distinct stages for further analysis. For example, we quantify the time and the distance between successive steps. There's considerable clinical value as a descriptor of functional status. So this is a well-known figure in the gait field. It shows how the elements of each step are broken down. And as shown here in blue, the step pattern of a disease-free individual is pretty regular. Below is the Parkinson's gait. So here we can see short step length, increased cadence, and shuffling, and this gait pattern is characteristic of Parkinson's disease. What about gait in Prader-Willi syndrome? So studies suggest that children with Prader-Willi have a gait pattern that's cautious and slow, and they have difficulties with initiation and termination, getting going and stopping. So gait difficulties in Prader-Willi could be related to problems with movement planning, coordination and balance, attention, and physical factors also play a role. The beautiful thing about gait is that it's so easy to measure. This means that it could be a very simple, straightforward clinical biomarker to be used to measure outcomes in trials. So traditionally, we would analyse gait using a mat embedded with electronic sensors or in a laboratory with cameras. These measurement techniques are gold standard and they'll always have their place. But what you get here is the white coat syndrome. The individuals may overperform, and the gait pattern, it's not really reflective of what you see on a day-to-day basis outside the lab. Also, they can only pick up short walks rather than long, continuous walks. So our data suggests that the longer walks are better for children in general because they reduce anxiety, and they're especially good for children with Prader-Willi syndrome. There's also important logistical issues to recognise. The mat is really heavy, and it's hard to transport, and it's hard to find space to do your experiments. And the gait lab is expensive to run, and there's very long waiting lists. So we want children with Prader-Willi syndrome. What we want is for them to be able to perform gait assessments in the home and the community setting. We want data collection to take place over a short walk to the park. We want to be able to perform assessments on rural patients in their own environments. And we want to link gait analysis with telehealth in the future. We want to reduce the very high appointment load. So to do this, we've been working with shoe-worn digital gait analysis sensors. These are fantastic little devices that attach to the shoe. So either on the side of the shoe or they're on the top. And we turn them on through a Bluetooth remote control. And then off we go for a walk. These are objective, repeatable, simple, and real world. But the second most important part of this work is around the feasibility and the robustness of data collected in continuous walking assessments. This is an assessment where the participant just goes for a walk with the assessor for a few minutes. They might stop or get excited, and people walk by. And this is what happens in the real world. So this is very different to the traditional modes where the child is asked to walk a short path or around a pole and back. And we need to do this for a few reasons. First, data quality. So the longer walk provides more data. And we need enough data to look at a very important variable called variability. So these are the fluctuations in stepping over time. This is one of the most robust markers of neurological function, probably the most viable candidate as a gait biomarker. And it's very sensitive to developmental level. Second, inclusivity. We need assessments that work in all children with Prader-Willi, including those that struggle with instructions. So it has to be simple. And scalability. Prader-Willi is a rare disorder, so we need paradigms that can be scaled out to the international stage. Our team here in Australia is working hard to tick the boxes needed to demonstrate robustness of gait analysis as a novel innovative biomarker for Prader-Willi. Now, this is not a small task. Here are some of the main issues that we've been addressing in our research. Science and purpose. We're drilling down to the different variables. We think the gait variability would be important because of its link with the cerebellum, but there's more work to do. And there's a lot of variables to look at. The type of biomarker that we think would work best would be a monitoring biomarker, so used against other tools. We've demonstrated feasibility of the long continuous walking assessment. We've done quite extensive work on analytical validation and have multiple papers near submission in this area. These include studying technical agreement with a gold standard system, establishing a normative data set, and developing algorithms to target steady state gait. And our work on clinical validation is preliminary, but we have some interesting data supporting larger studies. So firstly, just briefly, I'm going to talk about some of the analytical validation work. Our agreement studies and the walking bout selection algorithm, both are very important. So in this paper, we have attested agreement between the Physiolog gait up sensor and the gait right electronic walkway. We did this in 30 typically developing children aged 12 to 15. We have very good results. So we found that the sensors have an almost perfect level of agreement with the walkway, and near perfect for stride time, and near perfect for stride velocity and stride length. And there's the bland Altman plot. Second, we are working hard to separate the wheat from the chaff with a targeted walking bout selection algorithm. None of this will work if we can't manage our data exports. They're currently too complex and overwhelming for non-experts. So most sensors will come with the sort of black box system, which you can typically take out the part of the walk where the child is walking, or the child is turning, sorry. So it just gives you all the steps that were straight. For example, here, the x-axis is the cycle number, roughly every second step. And the y-axis is the foot turning angle, as shown here. The system, the gait up system, will remove all the data where the foot angle is greater than 20 degrees. And this looks good. It cleans the data. But this needs to be developed much more to work for Prader-Willi and other groups with neurodevelopmental issues. Children pause, children get distracted, and this is not always picked up with the turning angle data. So instead, we're taking the challenge on board, and we're working to find within this data the steady state gait. And we're working on an algorithm to do just that. What does this mean for the researcher and the family? It means we have confidence in the data. And it means that we can target motor control by overcoming the influence of distractions on gait results in these more fluid assessments. So we've tested this walking bout selection algorithm by examining agreement in variability between walks across different experimental setups. So we did this in children with Prader-Willi and controls. So first here is the short walk. This is a little boy with Prader-Willi. He is 8 years old, UPD, walking a 15-metre track pole to pole 3 to 4 times. And then here is our continuous walking assessment, which was performed at the hospital. And this is just a walk for 2 minutes. In this figure here, you can see stride time variability. So each little dot represents the variability score for each participant. First here, so S0 is the code we've used for. So this is what happens to the variability data when you only take out the turning angle. For some participants, the variability is through the roof, and I don't think that that is picking up motor control. It's picking up distractions, pauses, all sorts of other things that were missed with just the standard cleaning process. Then S1, this is what happens to the data when you take out the turns before and after turns. So take out the steps before and after the turns. So it cleans it up quite a bit, but you still get participants with inflated variability data. With our model, this tightens the data up quite a lot, and the variability in the short walk is pretty similar to the variability in the longer continuous walk, and that's what we want to target. That's that child's most normal gait. So that's the gait that we think is reflecting their motor control system. It's tricky, but it's extremely important. And here's a bland Altman plot showing agreement in variability between these two different walks after using our reductionist algorithm. This has a very, very powerful capability because it removes the need for protocols and instructions. Clinical validation. So here's just some quick data from a Prader-Willi pilot study. We're looking at intergroup differences and relationships to clinical phenotype. We collected data from 21 control children and 7 with Prader-Willi. We were able to match the groups on age, BMI, and gender or sex, but there were some differences with weight, height, IQ, and Prader-Willi had much higher parental reported levels of autism symptoms. So here is video 1. This is the young 8-year-old boy with a UPD, just performing the assessment. He's walking with the assessor. All the children have the option of bringing their parents, but none of them wanted to, which was interesting. I guess wanting to express some sort of independence there, which is great. Anyway, so that's our boy. His gait is looking pretty regular, pretty good. Here is the boy with deletion. He was quite different. So he was quite insistent on having his arms crossed, and we had to engage in conversation to reduce anxiety there. And as you can see, when he uncrosses his arms and starts to relax, this stereotypical motor pattern emerges. So he's bouncing a bit there. So intergroup differences in stride time and cadence were studied in this pilot investigation. With very clear results here showing that the Prader-Willi group, the stride time, so that's the time from step to step, is slower for Prader-Willi children compared to the typically developing group. Similarly, cadence was slower, so they're slower and less frequent. But interestingly, we also found that they had a longer stride length. So they're slower, less frequent, but their steps are longer. So they seem to be compensating for that slowness by lunging forward a bit more. We also looked at variability. So this is stride time variability. Variability was calculated as the coefficient of variation. And we found significantly increased variability for multiple gait parameters: stride time, cadence, stride length, heel strike angle. So overall, the Prader-Willi group was less consistent from step to step on lots of different variables. And here we have stride time variability data. And, you know, even the boy that looked pretty good was much, much more variable than the controls. And I forgot to mention before, so the solid circles are males and the open circles are females, and seems to be a bit of gender difference there, but a very small sample. We also looked at possible relationships between gait patterns and parental reported conduct problems and emotional symptoms. And here we use the Strength and Difficulties questionnaire. So here is the data for stride time on the y-axis and conduct problems on the x-axis. Very small sample, and I really want to stress that we should need to take this data with caution. And yeah, so it suggests possibly slower stepping is associated with increased conduct problems. And then again, the flatter stepping was found to maybe be associated with increased emotional symptoms. But again, very small sample size. Conclusions. So, gait analysis with gait-up physiolog wearable sensors. It was a feasible experiment outside the lab setting in children with Prader-Willi syndrome. But application of the algorithm to identify that steady state gait was necessary. We found intergroup differences in stride time and cadence, and this demonstrated a slower gait in the Prader-Willi group that seemed to be compensated by a longer step length. Possible relationships between gait patterns and parental reported conduct problems and emotional symptoms. Future directions. We need to follow this up with more studies and take the necessary steps to validate gait in this approach to gait analysis as a biomarker. And we need to get regulatory approval to use this tool to monitor responses in drug treatments. We need to trial it as an outcome measure in natural history studies. Very importantly, we need to assess the feasibility and the acceptability of parent-led home and community assessments, because this is the ultimate goal provided by wearable sensors. We need to perform comparisons to our normative data set. This is important to untangle complex relationships between age and gait. And finally, just as a final note here, this is the technology that's available. It's pretty good, but it won't be long before we have things like this, clothing with technology embedded into the garments. These are being developed now. It won't be long before we have technology like this, also at prototype stage. So these are smart clothes that have technology embedded within the textiles. And then it won't be that far off that we have electronic e-tattoos that have the sensors put directly on the body. So I have no doubt in my mind that this is where the future is headed. And it's exciting because it has the potential to ease the testing burden for children with Prader-Willi syndrome and their families, but without compromise to data quality. So finally, just acknowledgements. I really need to thank the diagnosis and development team, my fantastic supervisor and mentor, Associate Professor David Godler, my mentor, David Amor, colleague Emma Baker, and of course, Parent Date, who did all of the assessments, which was absolutely wonderful with the children, and our collaborators, and of course, thank you to all the families that participated in our research and our funding. Thank you very much.